Hello, my name is George Adams and today I'll be talking to you about my project, which is determining the effects of airborne particles on immune and epithelial cell function. Particulate matter pollution poses a significant risk to human health, with approximately over 3 million deaths a year globally attributed to outdoor pollution. It has been linked to several cardiovascular and pulmonary disorders, as well as diabetes and several types of cancer. Particulate matter is made up of biological components such as pollen, fungi, bacteria, as well as non-biological components such as soot, diesel exhaust particulates, and transition metals. Now, how these components interact with the immune system and lead to inflammation is not fully understood. That's where my project comes in. The aim of this work was to expose lung epithelial cells to chemicals derived from diesel exhaust particulates. As diesel exhaust particulates are omnipresent in the environment, they provide a good model to assess airborne pollution. 3 chemicals were used, but for the purpose of this presentation, I'll just be talking about naphthol-quinone. Now, cells were exposed to naphthol-quinone with and without the presence of a bacterial stimuli, LPS, and we assessed the physical and immune responses. Now, as we can see from the top 2 graphs, naphthol-quinone induced cell death at high concentrations, but not cell proliferation in a dose-dependent manner, with no effect from LPS. Whereas naphthol-quinone induces release of reactive oxygen species and expression of several pro-inflammatory cytokines such as IL-6, IL-25, CCL2, and IL-33. But this response was reduced in the presence of LPS. Now, our key findings show that naphthol-quinone, which is derived from DEP, does induce expression of pro-inflammatory products. And these products could promote the recruitment and activation of immune cells and inflammation. However, we predicted that the presence of bacterial stimuli would potentiate the response seen from chemical stimulation. However, this seems to have a negative effect with the presence of bacterial stimuli leading to reduced inflammatory response. Now, in order to further investigate our work, we plan to investigate epithelial cell signaling pathways to see what is occurring within the cells leading to the inflammatory response we are seeing, as well as sampling of ambient environments in order to compare the data we have from our models to real-world comparisons in order to see if the results are the same. Thank you for listening.